Hello student, I am Dr. B.K.Barun from Medico, Patna, and the today class is nucleic acid. The term nucleic acid was given by scientist Mixture, and the nuclein term was given by Altman. And nucleic acid consists deoxyribonucleic acid, that is DNA, and ribonucleic acid, that is RNA. So DNA and RNA is nucleic acid. The nature of DNA is acidic because it is deoxyribonucleic acid, and the charge present on DNA is negative charge. So DNA neutralized with the histone protein. So DNA attached with histone protein in eukaryote, because DNA forms DNA, DNA forms then RNA forms protein. That is central dogma. So after formation of DNA to DNA or RNA or protein, then histone protein binds with the DNA, because histone is basic in nature in eukaryote. Histone present only in eukaryote. So eukaryotic DNA is combined with histone protein. So this histone combined with DNA and it inactivates the DNA after protein synthesis, translation or transcription or duplication. It is formation of DNA to DNA is called duplication or replication, and formation of DNA to RNA is called transcription. And sometimes RNA form DNA, that is cDNA, complementary DNA, mainly in the retrovirus, AIDS virus, that is the reverse transcription. And formation of RNA to protein is translation. So the overall process is called central dogma, and formation of RNA to cDNA, that is reverse central dogma. The central dogma was given by scientist Crick, and reverse central dogma was given by Temin and Baltimore. So it is also called Teminism, formation of cDNA, that is from RNA to cDNA, that is reverse transcription. An enzyme utilizes reverse transcriptase or RNA dependent DNA polymerase. And formation of DNA to DNA, the enzyme is DNA polymerase. It may be DNA polymerase first, second, third, and eukaryote, that is alpha, beta, gamma. So this DNA has negative charge and move towards the anode, and DNA combined with histone protein after duplication or replication. But in prokaryote, histone is absent. So the prolamine, that is type of protamine, which is basic in nature, combined with DNA after replication. This is difference between DNA and RNA in prokaryote and eukaryote. In eukaryote, DNA attached with histone protein, but in prokaryote, DNA is naked because histone is absent. But at the place of histone, other basic things, that is prolamine, that is a type of protamine which attached with the DNA. And eukaryotic DNA is double stranded and helical and linear, but prokaryotic DNA is circular, double stranded, and no any free end. But in eukaryotic DNA there is a free end, 5' to 3' or 3' to 5', that is anti-parallel DNA, mainly in balanced DNA, that is bDNA. So this is difference between prokaryotic DNA and eukaryotic DNA. Other DNA also present in prokaryote, that is called plasmid, which is extranuclear DNA, which is different from the main DNA. Main DNA in prokaryote is called genophore or nucleoid, because nucleus is not surrounded by nuclear double membrane. This is false nucleus, and the DNA is called genophore. And this genophore consists the circular and double stranded, but eukaryote DNA is linear and helical DNA. And this helical DNA was given by scientists, that is Watson and Crick. So Watson and Crick model DNA follows the Chargaff's rules, and RNA not follows the Chargaff rules. So this is difference between DNA and RNA also. RNA not follows the Chargaff rule because RNA is a single stranded. It may be L-shaped RNA. Mainly L-shaped RNA is mainly three-dimensional RNA, that is 3D. And the RNA is 2D model was given by scientist. Cloverleaf model was given by Holly. That is two-dimensional model, 2D model. So DNA is different type such as bDNA, pDNA, cDNA, eDNA. But RNA is messenger RNA, transfer RNA, ribosomal RNA, small cytoplasmic RNA, hnRNA, heterogeneous nuclear RNA. So these are the different type of RNA. The most common RNA and the maximum percentage of RNA present cell is called ribosomal RNA, around 70 to 80%. And transfer RNA is around 10 to 15%, and messenger RNA is very less, that is the 3 to 5% overall the cellular RNA. So these different type of RNA. RNA is a genetic material and DNA is also genetic material. If DNA is a genetic material, then RNA is non-genetic RNA, such as messenger, transfer, ribosomal RNA. And if RNA is a genetic material, mainly in virus, then DNA is absent. So some virus, DNA is a genetic material, but in some virus, RNA is a genetic material. Such as RNA genetic material was given by Frankel and Conrad, and DNA is a genetic material was given by scientist Griffith, Hershey and Chase. That is the isotopic study, and the Avery, McLeod and McCarthy experimentally proved that DNA is a genetic material. And the property of DNA is denaturation. It is important property of DNA. And DNA absorbs the 260 nanometer wave ultraviolet ray light. So this is also property of DNA. And hyperchromic effect of the DNA is also a property. So the different type of DNA and different type of RNA. Overall DNA RNA is a nucleic acid. Thank you.